Morning and welcome back to Chem 1804. I'm Dr. Hathcock. Dr. Alaban is controlling the camera today. And we are going to do fermentation of sugar. And the reason you guys are taking a look at this is if you were in my 1802, we did a lab working with eggs to simulate the biology and the chemistry behind cells, and we use the egg and the why am I drawing a blank? We use the egg. Help me out here, Dr. Alaban. We use the eggs.
Osmotic.
Egg cells.
Semi-permeable membrane?
That's the word,
okay.
Semi-permeable membrane. Well, I don't know why I was drawing a blank there today. We use the semi-permeable membrane to simulate the barrier between the inside and the outside of a living organism, and how we could show the difference between how that membrane allowed diffusion to show you osmosis, specifically whether or not if you had an isotonic or a hypotonic or a hypertonic solution, and what would happen between those membranes and what happened with those solutions to simulate a cell in the body. Now we're going to extend that in this class, and we're going to talk about the extension of this in how that applies to biochemistry. So we're still along the life science side of it, and how biology and chemistry still work together with the fermentation of sugar. Now, many of you are possibly athletes, and so as an athlete, you know, or may have experienced as you were running or exercising, extreme exercising, and hit something that athletes often call the wall, where you've used all the lactic acid in your muscles and you can't go any further. I mean, you are at your stopping point. There is nothing left. And that's basically where you have used up all the sugar in your muscles, and you cannot produce any more ATP to do anything else. And so that is basically where you are out of oxygen, you are out and you can't go from the aerobic or to the anaerobic. There is just nothing left. So athletes often term that the wall, and you often just say, I'm done. So in biology or biochemistry, we call that that's part of the Krebs cycle, and that's where you are actually transforming sugars and oxygen, nitrogen, and you're going through the entire Krebs cycle. And I'm not going to go into the Krebs cycle here, but basically your energy source, which is ATP or adenosine triphosphate. And there are two possibilities to do that. You can either do it with oxygen or without oxygen. And so in this particular lab, which is the fermentation of sugar, we are actually going to simulate that. So we're going to simulate the Krebs cycle more or less, and in the fermentation of sugar with oxygen, and we're going to do it without oxygen. And so that's basically the idea behind this particular lab. So it's an introduction, although a very basic introduction, to the biology side of chemistry, which is the course on biochemistry, which is often completed after you've taken general chem and organic chemistry. So we're going to look at fermentation, which is the anaerobic side of ATP process here. Anyhow, so you will have, of course, the kit specifically for this lab, and then you had the general kit, which came with the supplies here that I have laid out for you. Now, I've prepped part of what you needed to do, which was I made up my yeast solution, 35 degrees Celsius, and my 2 grams to 125 mils of water needs to go in mixed up. I've also have my 35-degree water so that I can create my gas collecting chamber. And with the fermentation of sugar, there's some background information. You will be looking at plain old sucrose, a lactose monohydrate. You'll also be looking at a D-glucose anhydrous, which is basically a waterless sugar. And it's a very simple process that you do over a course of 60 minutes, and you're going to test them, all of the different samples that you are given, and each trial runs 60 minutes. So this is actually a rather long lab. And so we are going to actually run one sample, and we'll actually run the sample, but we're not going to record all 60 minutes. We'll run one sample the first 15 minutes. We'll pause the video and come back with the results and show you the calculations. And so there's a video that shows you a different setup using a graduated cylinder. I'm going to actually show you the setup using the, what do they call this?
Centrifuge tube.
Centrifuge tube. Thank you very much. And I've gone ahead and pre-weighed out my 1 gram of sugar. I've already moved my yeast over to my bottle, set up my tubing. You get 3 sets of tubes, you get 3 bottles. I've hooked up the tip of my tube, and it comes with visuals and figures. So I'm going to go ahead and I filled up my centrifuge tube. With 35 degrees water, and it gets a little bit messy, but I purposely filled it to the top so that when I flip it over, I'm going to have possibly, hopefully, no air. So I'm going to quickly flip it over and put it in there. Now, when you look at these tubes, if you notice the reading, it starts at 1/2 ml at about halfway down the tip. The 1 ml mark starts at where you get from the tip of the cone to, I would call it, I guess, the horizontal point at which the cone then becomes level. So if you look at this, I'm going to record this at probably 1.25. So that's going to be the starting point at which my gas will begin. And the reason this is important is because my initial gas here for volume, it doesn't start at 0, it starts at 1.25 rather than 0 because I have an air pocket. So it's kind of like reading a ruler. A ruler is usually marked at 0, but I could start anywhere I wanted. But if I start here, I'm not starting at 0, I'm starting at 1, so that would be my beginning. So my beginning is actually at 1.25, and so that's what I'm actually going to mark as my initial starting point. So my initial starting point is 1.25 mils. Now, I'm going to be fermenting sucrose to start with. I pre-measured 1 gram. Now, we all know that yeast doesn't actually grow without some sort of food source. And if you're in cooking, or you've ever baked anything before, sugar is a food source of yeast. Yeast is also known to be an opportunistic fungus. It needs something to feed on. Sugar happens to be what yeast feeds on. Now we want to use, oh, here it is, a barrier. And the barrier happens to be mineral oil. Now it's really important not to mix the yeast with the oil. So you're going to put about a half of centimeter, half a mil, because remember cubic centimeter is the same as a half a mil. And so the best way to do that is to slowly layer it on top. Now if you notice, I'm using a pipette. Once I mixed my yeast with the sugar. And you should see a layer form on the top. A little more than a. Okay, then you're going to put your tube with the and begin to collect your gas. You'll measure this in 15 minutes. And we'll begin our time now. All right, an hour's gone by, and I have some values for you.
Occasionally I see some glare.
Okay, so I only did one of the three samples that you have. So we're focusing on sucrose. So initially I had 1.25 mils to start off with. After 15 minutes, we went up to 3 mils. After 30 minutes, we were at 3.5 mils. 6 mils at 45, I got lazy. I stopped writing my mils. At 60 minutes, we were at 7 mils. So to come up with a total, I took my final value, which was 7, and I subtracted my initial value. That put me at 5.75. Now let's make sure my values are correct. So 7 minus 1.25, that gives me 5.75 mils.
And that 5.75 is how many milliliters of gas the reaction produced.
So that's gas. Because what we're doing is we are collecting gas volume observed. So that's gas
that initial
volume.
The reason we take our initial volume of 1.25 is because that first air pocket was not produced by the reaction.
So that's what happened when we flipped over my centrifuge tube. Okay, so now in step 17, now this is off of page 9, I want to calculate the milliliters of carbon dioxide and the moles of ATP that are produced in each gram of sugar by the fermentation from my yeast and sugar that were combined. To do this, we need to use the gas volume that was observed. So there's a formula that you have in your activity, and it's actually given to you in step 17. Now it's also given to you in your background data, but it's easiest just to go to step 17. Now that formula that we're using, and I'm actually going to walk you through it, is, and we'll come back to recording our data in just a second here, is your start volume. Oh, sorry, I'm going to do it backwards. Well, actually, this works too. It's just the absolute value. But if you take your final volume minus your start volume, and what we found that to be was the 5.75 mils.
So it's the 7 minus 1.25.
Okay. We're going to divide that by our grams of sugar. Now, you're going to have a variety of values because nobody's perfect. I just happen to get 1.01. Okay. So I took 7 minus my 1.25, and I'm going to divide that by my grams of sugar. Now remember, this is mils of CO2 divided by grams of sugar, and because we're looking for moles of CO2 divided by grams times minutes, so grams of sugar times minutes, because that's how long we took. So grams of sugar times minutes, I had 1.01 grams of sugar times the total time my experiment took, which was 60 minutes. So I ended up with 5.75 divided by whatever 1.01 times 60. Now remember, you have to do the top first and then 1.01 times 60 first, then you can do the total division. So you get 60.06 for the bottom. So I end up with 0.0957. And if you go out further, and it doesn't tell us how many significant digits to go out, then you're going to divide all of this by
24,000.
24,000. Is it divide by 24,000?
Yes.
Because on page 4,
look at page. Now you're looking at step 18.
Right, step 18.
Over here.
Oh, I'm looking at the wrong page.
Now it's 24,000, not 24.
Oh, right. Okay, so this gives you your milliliters of CO2 per gram.
Per minute.
So 0.0957 CO2 per gram.
Minute.
Now in step, what?
Minute 2.
Yeah, thank you. Grams per minute. Now in step 18, that will give you your moles. So we're going to take this value and we're going to divide it all by 24,000 to find moles. So 0.0957, which is CO2, divided by grams per minute, and we're going to divide it by 24,000 because that's how many moles there are, and we will get,
you should get a very small number if you end up.
holy cow, we get 0.0001, 1, 2, 3, 4, 5.
I think you're looking usually about somewhere between -6,
3, 9, 8, 9.
about -6, -7 for if you're looking at scientific notation.
So that comes out to 3.989 times 10 to the 1, 2, 3, 4, 5, 6. Negative 6th power of CO2 grams per minute mole.
Nope, moles of CO2.
Oh.
It should be moles CO2 per gram minute. And if you get something of times 10 to the 22nd, 24th, you've done something wrong with the calculation. So this needs to come out to a very small number.
Okay, now, so I have 3.989 times 10 to the negative 6th.
That's moles of CO2, not milliliters of CO2.
Oh, my writing's bad. Moles.
You do have 3 columns, so you will have to tell me the milliliters of CO2 per gram minute and moles of CO2 per gram minute, and then the moles of ATP per gram minute is a very easy calculation to do.
Now, in your background information, what it does tell you is that all of your equations are 1 to 1 to 1. If you have 3.989 moles of CO2, no, that one was mils.
No,
And then
0.9.
there was moles.
If you flip back over, it's 0.0957 milliliters.
Yeah, this one was mils here.
So that's your mils.
And then it was converting to moles. I was doing step 18 here.
So, yeah. So you've got 0.0. So that first one, what you've got there is you left off a box. So that, if you originally had milliliters there,
Right.
and that would have been the 0.0957.
Yeah, I know. I was going to fill this one in and then fill that one in.
Just change moles. The table's backwards.
Right, I was going to fill it in.
Okay.
I had it down. Too many cooks in the kitchen. Okay, so you have 0.0957 mils of CO2 and 3.989 times 10 to the negative 6 moles of CO2. So on page 4, you will notice that if you have 3.989 times 10 to the negative 6 moles of CO2, you also have 3.989 times 10 to the negative 6 moles of ATP, and 3.989 times 10 to the negative 6 moles of the third one.
You're going to calculate milliliters?
Yep. Mils, moles, and moles.
Yeah.
All right. I think that's it. We'll see you in the next one.